Hi guys, this is Jennifer from El Guru. Today we are going to see about the fundamental unit of life, part 2. In the part 1, we have seen about what is cell, how it was discovered, and various contributions of scientists, what is a unicellular organism, and what is a multicellular organism, and about cell diversity, cell shape, cell size, and cell numbers. And also we have seen about nucleus. And we have seen about the cellular membrane, cell wall, and other things. So today we are going to see about the cytoplasm, cell organelles, and cell division. Come, let's go through our slides. Now we are going to see about cytoplasm. So what is cytoplasm? Cytoplasm is nothing but the fluid which is filled inside the cell. So there will be some fluid substances which are present in the cell, and this fluid where you can see this other cell organelles are suspended. This fluid is said to be cytoplasm, and the area is said to be cytosol. The cytosol encompasses the filaments, ions, proteins, and macromolecular structures. And other organelles are also suspended in the cytosol. So, in the eukaryotic organism, the cytoplasm also doesn't include the nucleus because the nucleus is having a clear nuclear membrane. But in prokaryotic cell, there is no nuclear membrane. So the cytoplasm also includes the nucleus. Now, here you can see the diagram of the cytoplasm. Here in this diagram, this part, this white color part is said to be cytoplasm. Here you can see in this part, this is a fluid, and you can see this, all the other organelles are suspended in this cytoplasm. Now let's talk about cell organelles. So in our body, in human body, and in all living creatures, there are so many organs present, and different organs will perform different functions. For example, heart will pump blood, and bones helps in the bone systems, and different muscle systems are present, and also kidney and liver and various organs are present, and they perform different functions. In the same way, inside the cell, this cell organelles behave like organs. They are said to be small organs. So they are found embedded in the cytosol, and they will have definite shape, structure, and function, and they will also differ from its functions. So now let's see some of the cell organelles. Now let's talk about endoplasmic reticulum. So it is absent in prokaryotic cell and mature RBCs of mammals. So it occurs in 3 forms. The shape of the endoplasmic reticulum occurs in 3 forms. The first one is Cisternae. It is like a sac-like structure. The second one is vesicles, and third one is tubules. So tubules is tube-like form. Now let's talk about the types of endoplasmic reticulum. There are 2 types of endoplasmic reticulum. The first one is rough endoplasmic reticulum. The second one is smooth endoplasmic reticulum. So rough endoplasmic reticulum is named because it contains the ribosomes in its surface. Because of this, it is said to be a rough endoplasmic reticulum. The other type is smooth endoplasmic reticulum. In this, there is no ribosomes present. So therefore, it is called as smooth endoplasmic reticulum. So let's see about the functions of this endoplasmic reticulum. They help in the intracellular transporting. So inside the cell, the materials are transported, the organic materials are transported from one part of the cell to other part of the cell with the help of endoplasmic reticulum. So this smooth endoplasmic reticulum helps in the formation of lipids, in the synthesis of lipids, and the rough endoplasmic reticulum helps in the synthesis of proteins. So since they are involved in the synthesis of both proteins and lipids, the process is said to be biogenesis. So this is the structure of endoplasmic reticulum. Here you can see the rough endoplasmic reticulum. Due to the presence of ribosome, it is said to be rough endoplasmic reticulum. Here you can see the tubule forms where it is said to be smooth endoplasmic reticulum because it doesn't have any ribosomes in it. Now let's talk about Golgi apparatus. So the Golgi complex was discovered by Camillo Golgi in the year 1898. So it occurs in all eukaryotic cells, but it is absent in the prokaryotic cells. So this Golgi apparatus consists of a set of membrane-bound fluid-filled vesicles, vacuoles, and flattened cisternae. So what is the function of this Golgi apparatus? So this Golgi apparatus helps in packaging and dispatching unit of cells. It is said to be the packaging and dispatching unit of cells. It packages the organic material, and it takes to different organelles, and it also dispatches the materials. So it is also involved in the cell wall, plasma membrane, and lysosome synthesis. Okay, so it is also helps in the formation of cell plate during the cell cycle. Now this is the structure of the Golgi apparatus. Here you can see incoming transport vesicles. So the materials which comes in are taken inside and it is dispatched out through the outgoing transport vesicle. Next one is lysosomes. These lysosomes are dark and spherical in structure. The single membrane bound sacs containing several digestive enzymes. So it contains different digestive enzymes in it. And it is responsible for digesting and breaking down of all organic material. So these lysosomes are formed by the Golgi complex. And they are also found in the cytoplasm of eukaryotic cell, and it is mostly in animal cells. And this helps in the intracellular digestion. So during starvation, what happens? The cell digests its own organelles through the lysosomal enzymes, and this process is called as autophagy. And this also digests, it keeps the cell clean by digesting the foreign material which is entering into the cell. So sometime the cell will destined to die. After it is aged, it will destined to die. During that time, this lysosomal enzyme will digest the cell, and this process is said to be autolysis. So because of that, lysosomes are also called as the suicide bag of the cells because it's causing its own destruction. So it is said to be suicide bags. So this is the structure of lysosome. Here you can see there is a membrane, outer membrane, and also inner middle lipid layer is also present here. And there is also membrane transport protein is present. And inside this, there is a mixture of hydrolytic enzymes. The next one is mitochondria. Mitochondria is also said to be the powerhouse of the cell because it gives energy to the cell. So it is present in the eukaryotic cell, but it is absent in the prokaryotic cell. So it is a double membrane organelle. So the outer membrane is very smooth and the inner membrane is a fold forms, and it is said to be crystal. And the crystal consists of F1 and F0 particle in it. And also there is a matrix which is filled inside the ribosome, and it is this matrix is made up of respiratory enzymes and circular DNA in it. So as I told you already, this mitochondria generates ATP, that is adenosine triphosphate, which is the powerhouse. So it gives the energy to the cell. So hence it is called as the powerhouse of the cell. So this mitochondria will have their own ribosomes and DNA. So they can also synthesize their own proteins. So and they are called as self-duplicating units. So they are said to be semi-autonomous organelles. So this is the structure of a mitochondria. This is the outer layer, which is a smooth one, and there is an inner folded layer, which has F1 and F0 complexes in it. And this folded layer is said to be crystal. And there is a matrix where you can find the digestive enzymes, and also ribosomes and circular DNA is also found in this mitochondrial matrix. Now we are going to talk about plastids. These plastids are present only in the plant cell. So it is characterized as chromoplast and leucoplast. Chromoplast is the colored plastids and the leucoplast is the colorless plastids. So this plastid contains the chlorophyll pigment, which is also called as chloroplast. This chloroplast plays an important role in this photosynthesis in plants. This chloroplast also contains the yellow or orange pigments in it. So this leucoplast, it is very important. It is very important because very important materials are stored in this leucoplast. For example, starch, oil, and protein granules are stored in this leucoplast. So this plastids look like mitochondria, but the function is not similar. But the shape and everything is similar to that of mitochondria. So this also, like mitochondria, plastids also present. Plastid also possess its own DNA and ribosomes in it. This is the structure of a plastid. Now we are going to talk about the vacuoles. So these vacuoles are commonly the storage sacs, and where you can store the organic materials are stored in either solid form or in liquid form. It is stored. So here in animal cell, the vacuoles are very small, but comparatively it is very big in plant cell. So in plant cells, these vacuoles are filled with cell sac. Because of this, it is big in size, and also it provides turgidity and rigidity to the cell. Because of this vacuole, the cell is strong and it gives strength to the cell. Now, this is the important part of this chapter. So here there is an overview of the cell division. So what is cell division? Where the parent cell gets divided into two or more daughter cells. This process is said to be cell division. So this is one part of cell cycle. So there is a big cell cycle occur, and this cell division is one part of this cell cycle. So what happens here? The newly formed daughter cells will themselves divide and grow, giving rise to new cell population. So this is said to be cell division. Okay, where there are so many cells are formed from a single parental cell. So in other words, we can say a single cell gives rise to millions of cells. So there are different phases here: interphase, prophase, prometaphase, metaphase, anaphase, and telophase, which is also called as cytokinesis. So this will be the about detailed about this you will be seeing in your future classes. So that cell division is of two types. So it is majorly of two types. One is mitosis and the other one is meiosis. So what is mitosis? Here, this is a process where the parent cell will form the daughter cell, will divide to give rise to the daughter cell, which is a exact replica of a parent cell. So for example, mitosis, which is observed in almost all body cells. For example, eyes, skin, hair, and muscle cells will divide in this manner, where the parent cell will divide into daughter cell, which will be, the daughter cell will be exact replica of the parent cell. But in meiosis, the cell division will occur. So it forms sperm or egg cells, and they doesn't produce the identical daughter cells like mitosis. It doesn't produce identical, but it gets divided into two daughter cells, but it won't be an exact replica of its parent cell. And one more type of division which occurs in unicellular organisms such as bacteria. So this type of replication is said to be binary fission, where you will find the DNA gets replicated into two, and then cytokinesis occur and the cell will divide into two. This process is said to be binary fission. So here it is a diagrammatical explanation of mitosis and meiosis. So here you can find a single strand of DNA present, and during replication it forms a double-stranded DNA, like X chromosomes it forms, and it gets divided into two. Like cell division, it gets divided into two daughter cells, which is exact replica of the parental cell. It doesn't look like a this diploid manner. It is not looking. It is haploid in nature, which is looking exact like a parent cell. But if you see here, it is the single-stranded DNA. During replication, it becomes double-stranded, like a hex chromosome. And this divide, each cell gets the pair of double-stranded DNA to each of the cells. And here you can see the cell division. In the cell division, this gets into two daughter cell, which is a haploid in nature, where it is single-stranded, where it equally shares two strand of DNA. So this process is said to be meiosis, where it is not looking exactly like a parent cell. So here binary fission. First, the chromosome, where DNA replication takes place, and it will divide into two. Once it is divided, after that cytokinesis happens, where cytoplasm gets divided into two to form two identical daughter cells. So these are the phases of cell cycle. So the first phase is interphase, and the other one is M phase or mitotic phase. So in this phase, earlier it was thought that it is a resting phase, but new research says that it is a very active phase, where the things happens for the cells get ready for division in this phase. So it is said to be the active phase. Next one is M phase, where exactly the cell division takes place. Where in the interphase, the cell will get ready for cell division, and in the M phase, the cells will get divided. So there are two steps in this M phase. One is cytokinesis, the other one is karyokinesis. So the interphase will comprise three phases. First one is GO phase, that is resting phase. Second one is G1 phase, that is gap 1. Third one is S phase, that is synthesis phase. And then G2 phase, where it is gap 2, and it comes to quiescent stage. So in the GO phase, that is a resting phase, where the cell will neither prepare for itself for division nor it gets divided. So it is said to be in resting stage. Next one is G1 phase. Here the cell is metabolically active. It will grow in size, but it doesn't get divided. So the next one is S phase. In this phase, the cell chromosome DNA will get replicated into two. And the next one is G2 phase. Here the protein synthesis will occur. In the first one, in the GS phase, the DNA replication occurs, and in G2 phase, where the protein synthesis occurs. And next is quiescent stage. In this stage, it goes to resting again. That is, there will not be any further division. There will not be any replication or division. So it goes to an inactive stage, and this inactive stage is said to be quiescent stage or GO stage of cell cycle. Now we are going to see about M phase. So there are 4 stages of M phase, where prophase, metaphase, anaphase, and telophase. So in this 4 phases, there will be cytokinesis and karyokinesis, where the nucleus will get divided into 2, and also the cytoplasm will also get divided into 2. So here in this diagram, it is explained all the phases: prophase, prometaphase, metaphase, anaphase, and cytokinesis. So first there is a division of karyokinesis, where the DNA is divided, where the nucleus is divided into 2. And after the nucleus is divided, you can see the cytoplasm is divided into 2. So in this way, the cell division takes place. So the very brief explanation of, detailed explanation of cell division will be shown in our future slides. You can check for the detailed explanation in our future slides. Okay, that's it, student. We are in the end of the slide, and so if you have any doubt, you can mail us or you can contact us, and I'm sure when you actually read it in your textbook, it's a lot of study, but believe me, it is not that difficult if you can understand it conceptually. All the teachers at Learners Gurukul are always willing to help you. So So do let us know if you have any doubts or need some clarifications. In case of doubt, you can go through our presentation and can also listen again to our lecture. Therefore, make sure to take quizzes to see if you are ready to go to the next chapter. Thank you so much, students. Thank you.